Chromosome structure and how it is formed. What is it? The best way to understand chromosome is to look at it as a DNA fragments, DNA which is wrapped, important, that's the word, DNA wrapped around histones. These are histones, proteins, which are like knobs. And the DNA chain is or is wrapped around these histones. You can see here how it is wrapped. This is the knob or histone, and you can see how is it depicted here. There are two. So it is bound or wrapped around the histone proteins. That is the first step in chromosome formation, because the genetic material DNA exists normally as chromatin inside the nucleus of the cell. So cell membrane, cytoplasm, nucleus is normal state. But when it goes into cell division, before cell division or mitosis starts, the nuclear material starts forming chromosomes. And after chromosomes are formed, mitosis will start and the cell will divide. Cell division takes place. Cell division is the process by which the body increases the number of cells. So cell division takes place. So from this unwound, unwrapped, and uncondensed, these are the keywords. Uncondensed state, the chromatin or nuclear material is transformed into chromosomes by a process of wrapping around histones and condensation. Now these are brought together or closer, and further these chains are formed. You can see the wrapping around and the condensation. So condensation occurs by forming loops, because a loop can have, suppose you this is 1 centimeter of straight line. In the same space, looping helps. Serpentine loops formation can help accommodate maybe around 5 to 6 centimeters or even more in the same space, and that is a form of condensation mechanism we find in chromosomes. Not only is there So not only is there condensation, it is wrapped, and during the time of wrapping there is already some condensation happening here, and then so there is double condensation. So this is a histone, it is wound. So not just the whole DNA chain is first, as you can see, wrapped around the histone, and then those chains are themselves condensed by this process of serpentine chain formation, rather than being in a straight line or an uncondensed, unpackaged manner. This is packaging. So that is how we can see here condensation takes place. And this is called nucleosome. The DNA wrapped around histone is called nucleosome, N. Let's keep it as N here. And you can easily say that a chromosome is made up of not just DNA which is condensed, but nucleosomes. Then it will be more technical and appropriate way of describing the chromosome. So we have to the keywords. If you're a beginner, there's a keywords you have to remember. What are the keywords? One, DNA. Number two, histone. Together they are wrapped. That's the third keyword. And the fourth is nucleosome formation. Then it's just a matter of condensation, further condensation, and further condensation. More and more chain formation up to a particular thickness. You can see two, you can do 1, 2, 3. So multiple such nucleosomes are brought together, and those chains are arranged in this way, and doesn't stop like this. You can see that they themselves are further condensed. So there's a high degree of condensation which is going on here, and that all results in what is called packaging. Is packaged, genetic material is being packaged to form chromosomes, preparing it for the process of cell division. So we should know, why is it even formed? Why? So we answer the question of why, what, what is it, and how. So the what involves DNA, histones, nucleosomes, and the how involves verbs such as wrapping and condensation. So wrapping and condensation, and the why involves for the purpose of cell division. Hope this introduction to chromosome was useful. This is very basic, and you need to grasp these things very, very rightly. Thank you. In the journal Cytogenetics and Genome Research, a study by the scientists involved a karyotype diagram containing the chromosomes, as we see here. So I'm citing the journal, and then we are going to the image. So this is the journal, Cytogenetics and Genome Research, and the topic is about chromosome painting and banding. To find out the structure, as we can see here, these are chromosomes pairs. So first thing is they are pairs, and this is not human. Okay, this is a different organism. But to demonstrate the point, to understand the structure, we can see that these are in pairs and there are bands. These bands are determined by segments with differing color or color density in the grayscale in this image. Okay, so that is the band or banding. And the chromosome itself, each one, this is the basic structure I would like to teach you in this section. So this is the short arm or P, this is the long arm or Q. So short, long. And the central part is called centromere. Centromere is the constriction. Centromere. These are the P arm, short arm, Q arm or the long arm. So the basic structure of the chromosome consists of two arms, short arm and a long arm. The short arm is called P, the long arm is called Q. The center we have the centromeres. And then we come to what we see here, it's banding. There are areas where there are segments, and these denote different areas where which are useful functionally. So that is called banding. Those are the bands, and this is how it looks. In humans, we do not have these two. We have 22 pairs of autosomes. So 44 autosomes, humans, and sex chromosome. So sex chromosome, normally you have to understand sex here as in gender, not the verb but the noun. So the noun of sex is gender. So gender determining chromosome XX is, as you know. So gender determining chromosome XX and the others are called autosomes. So autosomes and sex chromosomes. Okay. So humans have 22 pairs of autosomes and one pair of gender determining sex chromosome and autosomes. So these are 22 pairs. So there we have 44 plus here one pair PAIR. One pair plus 2, we have in humans a total of 46 chromosomes. So these are very basic things which we must be very, very thorough in our study of genetics. Next, we look at the human karyotype. So this image photo of stained chromosomes is called a karyotype. Karyotype. Next, we'll go to a human karyotype and see how it looks, how the 46 chromosomes looks. This is the human karyotype. You can see a male and female, and between these two I will draw a line to help you. You can see there are 22 pairs of autosomes. These are called, as we saw, autosomes. These are chromosomes which contain genetic information about all the cells of the body except the gender determining gamete cells or sex chromosomes. Okay, rest of the body, all the codes pertaining to the rest of the body are contained in the autosomes, whereas the sex determination is contained in these sex chromosomes. Gender. So male, as we saw, have XY. XY male and XX is female. What is the X and the Y? These are also chromosomes. So there is a X chromosome and there is a Y chromosome. So males have both X and Y chromosome. That is what makes a male, and that is what makes all the male hormones. And this will produce the male characters, sexual characters or gender characters, character gender, more gender character like development of penis, testes, and no breasts, or primary sexual, what are called primary sexual characters. So primary sexual characters are determined by the combination of X, Y, or X, X. So if it is XX, it is female, and this will produce hormones and growth factors which will produce the uterus, ovaries, vagina, etc., which are found in females and characterized females. These are primary sexual characters. Now we can talk about, you may think, what is a breast? What about breast? So breasts are secondary sexual characters. They are not mainly concerned with sex, though they are concerned with gender, but secondary sexual characters are not involved in baby formation or passing on of genes to the next generation. So these are secondary sexual characters. Breast is the secondary sexual character. Males will have mustache or whiskers. Mustache is a Now this brings us to the concept of ploidy. So ploidy is nothing but the number of chromosomes. 46 chromosomes is called diploid and 23 chromosomes is called haploid. So haploid is seen in gametes produced by the process. How is it formed? Haploid number 23 is formed by the process of meiosis, whereas diploid number 46 is formed by the process of mitosis. And this is found in all the cells except the gametes. So the normal cells of all the cells of the body have diploid number of cells 46, except the sperm and ovum, which have haploid number of cells. So these are the words they commonly used in genetics. Diploid and haploid, they use it repeatedly, and that means sperm, ovum, autosomes, there are somatic cells, etc. So you have to come to terms with those uses. Now we'll move on with ploidy. There are two more things I need to explain. In ploidy, we have polyploidy. It's nothing but the multiplication of the chromosome sets. Polyploidy is multiplication of chromosome sets. Polyploidy, and here we have 46. We can see here 46. 46 is euploid. Both the are normal ploidy. Euploid is normal ploidy. Polyploidy we have 46, XX, XY, plus either 23 or haploid, X or Y, or a 46, XX. You have to be very careful. This is how it will go in genetics, XX, XY, and this will result in what is called 46. So 22. So this is 69. 69 x x x slash 69 x y y, or other combinations of permutations of these two, xx and x, and all this can be added here. All this is possible. So this is one triploidy. It's called triploidy, and triploidy we have 69 chromosomes and 3 sex chromosomes. These are not normally found in humans. And tetraploidy we have 92 chromosomes and 4 sex chromosomes. So this is polyploidy. Okay, polyploidy. So 4 sex chromosomes. This is polyploidy, where we have multiplied or sets. You have 3 sets of haploid. 3 haploid sets form triploidy. Haploid is 23 chromosomes. So 3 haploid sets form 69 plus 3 6 chromosomes, whereas 4 haploid sets forms 92 plus 4 6 chromosomes. Okay, so this is an important concept called polyploidy. Next, we go to the concept of aneuploidy. So in aneuploidy, we have an additional number of chromosome. So we are now looking at 21. 21 trisomy is an example of aneuploidy. Now we're looking here. So this is a 21. 21 trisomy. The condition is called, so look, keep it in a field of view. So 21 trisomy is a disease produced when we have an extra chromosome in the 21st set. We have one more here, becomes 3, that's called trisomy of the 24 chromosomes. And here the ploidy is called aneuploidy. So we saw that euploid is normal, aneuploidy is abnormal ploidy. So this results in a disease seen in humans. It's also called Down syndrome, 21 trisomy. So we have seen the concept of karyotype, chromosome number, autosomes, sex chromosomes, inheritance through the gametes, haploid number of chromosomes from each parents which are inherited by the child, and also ploidy and abnormal ploidies. Mustache is a secondary sexual character in males. Similarly, breasts are secondary sexual characters in females. No breasts in males and no mustaches in females. Those are also secondary sexual characters expressed in a negative way. These are more for gender identification. Now we come to the gametes. The gametes are nothing but the sperm and ovum produced because of the action of the sex chromosomes or a gene expression. As a result of the gene expression of the sex chromosome XY or XX, you get the uh, testis or ovary, and the testis produces sperms, and the ovary produces ovum. Now they have some peculiar characteristics which we will discuss now. The gametes, unlike the autosomes in the rest of the body, do not have 46 chromosomes. So they have only 23 chromosomes. Gametes have only 23 chromosomes. It's very important to understand this concept. During mitosis in cell division, we get 46 chromosomes in every cell. And gametes have an extra process, special process called meiosis. And because of meiosis, the sperm and ovum have only 23 chromosomes. So 23 involves 22 autosomes and one pair or two and one sex chromosome. 22 autosomes and one sex chromosome. So that makes it 23. So now during the sexual intercourse, what happens? This verb as in a verb as commonly used during sex, we get sperm and ovum coming together. Sperm and ovum coming together in the body of the female, where the baby will be formed in the uterus. So in the fallopian tubes, you get sperm and ovum coming together, and one of and the process of fertilization takes place. Fertilization, and we get the zygote or embryo. Zygote, early embryo stage is called zygote, and this zygote contains as a new human being again 46 chromosomes. Okay, 23 from the sperm donor, is called the father in real life, and 23 from the ovum donor or the mother. So 23 from 23. So for that, for them to contribute equally, we need meiosis to produce 23 chromosomes in the gametes. So during sexual intercourse, the sperm and ovum, which are gametes, come together, and in the body of the female, in the fallopian tube, they fuse. This is called fertilization. And the fusion creates a new individual, human, called child or zygote. Human in the zygote form, single cell formed by a sperm and ovum coming together containing haploid number, producing a cell with diploid number. So the child has diploid number, half from the father and half from the mother. So this process is called inheritance. In genetics, you are inheriting the characters of father and mother, and you are the genetic expression of the combination of haploid genes of your father and your mother, and the outworkings of it in the given environment. So that is the importance of the haploid number 23 seen in the gametes only, only, only in the gametes, only in the sperm, only in the ovum we have haploid number because they have to come together and produce the individual with the diploid number of chromosomes.